Bonjour à tous et bienvenue à la première session de la série SciPol. Hello, everyone, and welcome to the first session of SciPol series of 2022. Before anything, I would like to start by acknowledging the land I'm speaking from, Richmond Hill, Ontario, which is the unceded territory and traditional land of Wenda, Haudenosaunee, and Anishinaabe peoples, whose presence here continue to this date. Today's session, Genomics on a Mission, is jointly organized with Genome Canada. I would like to thank my colleagues at Genome Canada for their work on this session. The session is in fireside chat format, and we'll have time for question and answers later. Should you have any question, please insert them in the Q&A box, and you can upvote those questions. Today's session titled is Genomics on a Mission. We would like to explore more about the role of genomics research in a wide range of sciences, such as climate sciences, but more importantly, its impact on policy and policymaking. And finally, about mission-oriented research, what it means and how it may impact us in future. I would like to invite you to read the editorial on this topic published in CSPC editorial pages 2 days ago by our distinguished panelists, which I would like to invite them to the virtual stage. And this is the time to introduce them. However, they need no introduction from me. Genome Canada's Dr. Rob Anand, President and CEO, and Dr. Catalina Lopez Correa, Chief Scientific Officer. Thank you, Rob and Catalina, for being with us today.
Thanks, Mehrdad.
Thank you. Great. Pleasure to be here.
Great to have you with us. So without any further ado, let me begin my questions. And in fact, we cannot deny that we are still in a pandemic. And the first question is relevant to the pandemic. So COVID-19 has brought the power of genomics to the forefront. Can you tell us a bit about the role that genomics has played in the pandemic and how this has informed policy? In particular, the new strains of virus. We have a new strain coming every couple of months, each with different clinical implications. And I want to know how genomics have played or have helped new policies for these new situations. Rob, why don't you start?
Sure, Mehrdad, I'm happy to start. And first of all, thank you to you and CSPC for hosting this today. It's CSPC is such a big part of the science policy landscape in Canada, so we're always happy to participate. Yeah, so of course, yes, we're still in the midst of the COVID-19 pandemic that has dominated not just our work but our lives for the last couple of years. And from the genomics lens, it's been interesting. Genomics has been a foundational piece of the overall response globally, and certainly also here in Canada, to both understanding and responding to COVID-19. I'll speak maybe kind of at the high level, and Catalina can speak maybe a bit more to some of the specifics around the variants and so on. But just maybe to go back to the earliest days of the pandemic, I think first of all, where we are in genomics today means that we were able to sequence the COVID virus, or the virus that causes COVID, very, very early on, which really kickstarted the medical response globally in terms of both vaccine development, which happened at unprecedented rates, as well as kind of both understanding the mechanisms of infection and so on. And that work was accomplished by scientists in China within about a week or so of the first isolation of virus. So that's an amazing demonstration of what genomics can do just in terms of providing information. In terms of responding, here in Canada, the response was immediate from people working in genomics who recognized how important it would be to not only survey for the arrival of the virus here in Canada, but to track its transmission, which you can do by looking and watching how various strains are circulating within populations, but also to watch for the emergence of variants of interest and variants of concern that may have different behaviors, which of course we've all gotten to know over time. So in the very earliest days, actually, there were groups of researchers and public health scientists across the country who started up very early conversations about how to start responding, how to start tracking the virus. And of course, Canada being a federated country, a lot of this was happening kind of at the provincial and regional levels. So we were able to come in as a national organization that works across an ecosystem to help, in a sense, bring those groups together, connect the dots between them, and then connect that into the broader federal response being led by the federal government and a variety of other national organizations, while at the same time maintaining that provincial responsibility and autonomy. And that was really through the creation of the Canadian COVID-19 Genomics Network, or CanCoGen. So this was a major viral response. At the same time, CanCoGen launched an important study of kind of the human genetic factors that contribute both to the varying responses we see in individuals to infection, as well as to factors that maybe contribute to long COVID and so on. So all this came together into a national network that built on the federated model we have here in Canada, with genomics at its core. And of course, one of the most important things we did was also to hire Catalina to come in and really lead that network nationally, which she's done more than capably since almost the start of the pandemic. So, I know maybe Catalina can speak a bit more to the actual variants and some of the details there.
Yeah, and thank you, Mehrdad and Rob. And I have to say that leading the Canadian COVID Genomics Network has been one of the most difficult things in my career, but also one of the most exciting positions I have ever had. And when I started, actually, we were very much focusing on the target we had in terms of sequences. We initially set up a target of sequencing 150,000 viral genomes and 10,000 human genomes, which at the time in April 2020, that seemed like, almost, incredible, a very, very ambitious target and ambitious goal. The reality today is that we have already sequenced 330,000 viral genomes, and we're getting close now to the 7,000 human genomes. So in terms of sequencing, the network has done really very, very well. But what is more important than generating sequences is how are we using those sequences? And back to your point, Mehrdad, to the initial question on the variants, as you all recall, we started to hear about those variants in December 2020. And December 2020, we started to hear about, initially we called them the, African South African variant or the UK variant, and then we moved into the alpha, beta, and the Greek alphabet numbering. But when the first variants appear, I have to say the network was already running for about 6 months. We were starting to build the capacity, but we were not really well prepared to face all these variants. So the variants was a big push for CanCoGen to really build the capacity and to start showing how we were using this data to inform public health and policy decisions across Canada at a provincial level, at a national level, and also at an international level. And when I'm talking here about policy decisions and public health decisions, we're talking about lockdowns, border closures, vaccine distribution, big important decisions in terms of public health that were guided and informed by genomic data. And I think that's totally unprecedented. We haven't seen historically moments or situations where genomics is really at the forefront of an important response for a province or for a country. So this, again, took us from just saying, oh, we're going to generate 150,000 sequences to we will inform and we are informing public health and policy decisions. Now, if we go, fast forward to December 2021, when we were all thinking, okay, we're good. I think we have this pandemic under control a little bit. And here we go, Omicron, the new variant, just before Christmas, when everybody was tired, nobody wanted to hear about more variants or anything. And I have to say that, thanks to the capacity we built across Canada, we started with only 4 labs that were able to sequence. Now we have 9 labs across the provinces that are able to sequence. We had only 3,000 sequences when we were in December 2020. We were close to 300,000 when December 2021. So we were in a much better position in terms of our capacity for genomic surveillance in December 2021 when Omicron appeared. And that is helping us make the right decisions, really inform what's happening with the distribution at a national level and international level. with Omicron. So again, genomics has been a critical component for the response, not only in Canada, but internationally to this COVID-19 pandemic.
Thank you for this very comprehensive answers. I know that there is so much to cover. I also appreciate. I have a list of questions. Appreciate perhaps shortened answers to the extent that is possible. We don't want to cut the content so much. We want to hear from you. These are all exciting stories, as Catalina and you mentioned that. I want to now focus a bit on policies as we look past COVID. What are some of the key relevant policy drivers we are facing? What are you seeing as major priorities in the government? The mandate letters published in December indicated the government priorities, but also broader in the science, technology, and innovation space that informs and shapes your work.
Right, still getting used to the mute button. So you ask us to shorten our answers, then you ask some big questions, Mehrdad, but I'll do my best. So, I think the mandate letters are obviously a really great way to sort of see very specifically what the policy kind of priorities will be for the period of time that the government is kind of in session. I will say though that I think it's important for us as a science community to both understand what those priorities are, which you can read. I won't repeat what's in the mandate letters, but more broadly, what are the broad kind of mega trends there, or the broader policy drivers? And as we think about, obviously COVID has dominated the space over the last couple of years, but it's not the only concern facing society, certainly right now. We need to be thinking about what we've learned through COVID about health more broadly in Canada. We kind of touched a little bit on kind of the challenges of kind of a fragmented system and building out capacity across the country. That's something that we'll need to be thinking about coming out of COVID as we think about other health things, whether that's antimicrobial resistance or what have you. We need to be thinking of, obviously, the next or the current global challenge we face around climate and climate change is clearly a priority for this federal government, but will be a priority for governments no matter their political background for years to come. And of course, science has a lot to add there. Food security, there's lots of issues, many of them tying into kind of issues around economic growth and economic sustainability. So those I think are going to be some of the major drivers. From our perspective, I think what's really fascinating is, I feel very lucky to be involved in the field we're in right now, because right now there's a confluence of factors that mean that the life sciences and kind of genomics in particular are super well equipped to actually address a lot of these drivers. And that really is kind of the confluence of the amazing sequencing work and the ability to generate data, but also the ability to manage and analyze that data. And then the development of lots of tools of engineering biology to actually address some of these big issues. So the technology and the science is fascinating. I think we're going to see a revolution in the life sciences in the next 20 years, like we've seen in the digital sciences in the last 20 years. But we will need to also take into account some of the underlying policy challenges around the fragmentation of the research ecosystem, our ability to connect kind of the fundamental research that is absolutely crucial into impact, whether that's economic impact or other impacts. We need to be thinking about talent and skills development. And we need to be thinking about, in particular, what we saw during COVID, issues around data sharing and the ability to actually move information around in a way that allows us to make the most of what we're actually generating through our research. So from a research perspective, those are kind of some of the big drivers I see.
Sabine, go ahead.
Yeah, maybe adding short, trying to make a short answer for a big question again, but adding a little bit to what Rob mentioned, I think CanCoGen, the Canadian COVID Genomics Network, has been a great example on how we can use genomics to address big, important global challenges. We are now thinking about our next venture. Of course, we will continue building the capacity for pandemic surveillance, genomic surveillance in Canada, and that's part of the work we will be transferring to the National Microbiology Lab. But we also see areas like climate change, as Rob mentioned, and sustainable agriculture, really environmental sciences, where genomics has a very, very important role to play, where genomics can help us understand what is happening in terms of our biodiversity can help us, and we have concrete examples of how genomics is helping us adapt to climate change, adapt our agriculture, our forest, and is also helping us mitigate climate change. And we do already have some projects funded by Genome Canada and the Genome Centers that are addressing some of these. The idea is to make it, as Rob say, I think where we are now is at a moment where we need a big push for coordination of the efforts. There's a lot of efforts that have been happening historically, but this is the time for us to coordination, the time for data sharing, and the time for aggregation and alignment to make sure we really have a concrete impact on those important global challenges.
Thank you, Catalina and Rob. Some of these issues that you mentioned, extend well beyond our borders. I appreciate the coordination that you mentioned nationally, but really these are global issues. And Genomics has a long history of international coordination, from the Human Genome Project in the 90s to the global genomics tracking of emerging COVID variants, right, recently. How is Genome Canada working with partners in other countries to ensure that Canadian research contributes to and also benefit from these international efforts? Catalina, you may go first.
Yeah, well, and I take again here first example of Cancogen, and I always say, as we know, the SARS-CoV-2 virus sees no borders. There's no borders for that virus. It's moving around the world freely and happily. And that's the way we have been trying to also address this challenge with genomics. We have been, I think internationally, it has been, again, unprecedented the way we are now sharing data at an international level. There's more than 7 million genomes sequence of the SARS-CoV-2 virus that are hosted in an international database called GSID. So it has been, Genome Canada has been actively collaborating with other countries, in particular the UK, the US, other countries in Europe, to learn about their strategies, to learn and to exchange best practices, to exchange protocols, and to exchange data. Of course, we can look at the positive side of things, but there are also many challenges on this international component of our response and genomics in general. And one of the big challenges is data sharing. As I mentioned, last year we only had a couple. End of 2021, Canada only have a few thousand samples that were shared in international databases. That was a big problem for us, because we wanted to be part of this international response, but yet we were not active enough in sharing the data and in really bringing that data to the forefront and integrating that data. So that's work we need to continue doing to be part, leaders in the international scene in genomics. So data sharing, I think, is the most important component here for us to be an international player.
I could maybe just really, I won't add much here, but to say, obviously you pointed out, Merida, that genomics has long been an international sort of field of science. I will just say that, internationalization is a good thing. Absolutely. There's no doubt about it. But too often when we think about international collaboration, we think about it among a pretty small number of countries. And we think about kind of, maybe sort of Western industrialized countries. I think as we're learning and hopefully continue to learn with COVID, as we move forward, we need to be making sure that when we think about international collaboration, that we're being much more inclusive. We're thinking about the global south, and we're not simply thinking about, something like data sharing, for instance, as an unalloyed good, and everyone should just put their data, into kind of universally accessible databases, but that we're making sure that the benefits that come from that kind of international collaboration are being equitably distributed and are accessible for parts of the world that are often excluded from these kinds of things. So I think the internationalization is absolutely crucial, but we do have a responsibility to be thoughtful about how we engage in it. There's no doubt.
Great, thank you. Now I want to switch to the mission-driven high-impact research. Genome Canada has embraced, a mission-driven approach in its research mandate. I want to know, what does this mean, and how is it different than before? And how does it fit within the broader discussion around, mission-driven research, as we often mentioned?
Sure, I'll maybe kick off here, Merida, just to say that, when we talk about kind of a mission-driven approach to research, it's really about really kind of organizing around what could also be kind of considered outcome-focused research. It's meant as a complement to what is obviously very important when it comes to like kind of a traditional sort of fundamental research or investigator-driven research, curiosity-driven research, which is really meant to be, I think, broad and exploratory, applied research, which is really dedicated oftentimes to addressing a specific issue. Mission-driven research, challenge-driven research is really meant to, in a sense, come bring researchers together to pursue a variety of potential approaches towards a common goal. It really starts with the outcome. It says, what do we want to achieve here? Do we, what is the outcome we're driving towards? And whether that has to do with COVID, for instance, is a perfect example where clearly therapeutics and vaccines and so on are major outcomes with lots of people pursuing different paths. We think about climate, for instance, there are many ways to try to get to net zero. But when in a challenge-driven environment, you're actually starting with that objective, and then you're bringing together a community to rally around really the creating the impact that helps to address that challenge. I think one of the real and of course, it's not just, this is what we're doing at Genome Canada. It's not just us, of course. Challenge-driven research has been around for a long time. DARPA in the United States is a well-known example. The Liberals have committed in the most recent speech mandate letters to set up a Canadian-type version of DARPA. Europe has a long history of mission-driven research. So it has shown that it can be really valuable and useful. I think one of the most important pieces for mission-driven research is that because you start with the impact, you can be very thoughtful about how those how the benefits are going to be not just created but then shared. This gets to sort of my last point around equity. I think in many ways we've thought about research from a bit of a kind of a trickle down kind of a philosophy that if you put in the money at the front end and you create great things, everybody benefits. I think it's pretty clear that's not necessarily true, that those benefits aren't shared equitably. So by having an impact-focused approach, you can actually start with the question, what are the impacts we're trying to create, and how do we make sure those are going to be shared equitably, and then work backwards from there in terms of the types of projects that you seek to support. So we're really keen on it. We've seen it work in many other cases, and it really fits well with the type of work we do in genomics.
Catalina.
Yes, maybe what I can add there a little bit on the equity and diversity. I think that's important here for genomics in terms of equity and diversity, and in terms of that capacity to deliver concrete impact to solve global challenges, and again, in an equitable and diverse way. And the way we're doing this, and we will be doing this at Genome Canada, is also through extensive consultation with stakeholders to make sure that we include those underserved group, the minority groups, the maybe isolated regions of the country in our dialogue from the very beginning. Once we start to think about the programs, we want to be more inclusive in our consultations, more inclusive in our dialogue. And again, this is a unique opportunity for genomics. And the challenge we have with genomics, as Rob mentioned, and not just in Canada, but internationally, is that it has been somewhat a little bit of an expensive technology, and also a technology that has mostly focused on having an impact in Europe, North America, but many regions, resource-limited regions inside Canada have not seen the benefits as many of the developed countries have seen. One important milestone for us starting this year was yesterday at Stanford. There was a publication where they say that they are now able to sequence a genome in 8 hours. This is a new Guinness record, and sounds like, a little bit of, like a media promotion for genomics, but it's actually a game changer. Think that the first human genome took 13 years to sequence. And now, in Canada, our fastest sequencing is about 1 week, maybe 5 days to get the sequence and analysis. But doing a whole genome and analyzing that data in 8 hours, again, that's a game changer. Reducing the cost, increasing the speed, and really providing the means to ensure the impact is for all of us. I think it's going to be a critical component of the work we will be doing around missions and focusing on big global challenges.
Thank you for that. I'm going to go to questions as they're already lined up, but I really would love to ask this last question, and I appreciate both of you. Very quick response to this, maybe 1 minute each. Genomics clearly, had a major role to play during pandemic and COVID. And the government has signaled, increased investment in genomics going forward, alongside other investments in biomanufacturing, life sciences, quantum AI, etc. Where do you see these investments taking us? And in your opinion, what do things look like 20 years from now? Especially, how can we make sure that everyone is benefiting? Who wants to start?
Catalina, do you want to kick off?
I can start. So I think, again, the use of genomics during this pandemic has opened so many doors for the future. We are seeing now evolutions of genomics into using genomics to monitor wastewater and be much more proactive on how we detect pandemics or pathogens even before individuals are sick. So I think we're going to move into a much more proactive and preventive in terms of health, use of the technology. We're going to move into using this technology and genomics merged with AI, quantum computing, and all this, this analysis of big data sets into, using that data to make concrete decisions about, where do we need to plant trees, how to bioremediate soil, how to advance precision agriculture. So I think, again, lots of the decisions, same thing as we've seen with our, use of genomics during the pandemic. I think that will extend to basically all sectors of the economy. With biotechnology, we're going to really transform the way we do agriculture, the way we do mining in this country, the way we're doing forestry. So I think the impact in 20 years is going to be, genomics is going to be a technology that's part of everyday life. And we sometimes do the analogy with the informatics revolution. Think of computers in the 70s. There were big machines in cold rooms, complicated. And think about what we have, the capacity we have in a cell phone today. Well, you don't see it, but, here is my cell phone. But, same thing with genomics. We will move into portable systems, cost-effective systems, easy to use. Sequencing a genome will not be a big deal, and we will be able to sequence genomes from every living organism here, and around the planet. That's going to transform how we do everything in our world.
Yeah, and it's hard to follow up on that. I mean, I think it is transformative. I think obviously there's the obvious kind of what we're learning right now, the near future around the ability to use mRNA technology and quick genomic sequencing to respond much more quickly to even flu season and so on. That the 8-hour sequencing transforms the game when it comes to personalized health and diagnostics. And so there's lots of ways, and then of course there's lots we can be doing in agriculture, as Catalina pointed out, forestry and natural resources. So, but to me, what's most exciting is the unknown. We know it's a powerful technology. We know that when it converges with investments being made in AI, in biomanufacturing, and so on, that new things will come forward. Catalina talked about the difference between the great big computers and your cell phone. Miniaturization, we can see that coming. But when I was just blown away that I could dial in on my modem and send an email, I never could have conceived of watching TikTok on my cell phone, right? So in the same way that there is an uncertainty to kind of how technologies might be applied, when they come together with creative, thoughtful people trying to solve big challenges. I think that in some ways the future is very much yet to be written, and I'm excited to be part of that.
Thank you, Rob and Catalina. Now I'm going to go to questions from the floor, virtual floor, and I already see a few of them lined up. But I want to ask my colleagues at CSPC to launch the first poll, and I appreciate the responses from participants. Please reply to these demographic questions, which help us better understand the audience and obviously better plan for future programs. And thank you for that. There you go. The question is launched. And meantime, I also want to mention that if you wish to submit your question in French, please do so. We'll read them in French, and if needed, translation, we will do that. And if you have questions, please put them in question Q&A box. If you want to generally comment and just express your opinion, feel free to do that in the chat line if you want to. But the questions I go through the Q&A box, which is ordered based on the upvoting, I guess. Okay, so the first question that I see here is from Patrick Blunden, and my advanced apologies if I mispronounced any name. The question is, the national work that has been done in genomic sequencing has been tremendous so far in Canada. What is your view looking forward in the next 2 years? Should we increase the sequencing work being done? Should efforts go more in bioinformatics of evaluations, or both? Who wants to take this question?
Well, maybe I'll just say a couple quick words, and then I can pass to Catalina, which is to say that the I think Catalina already summarized, the huge strides that have been made over the last 1.5 to 2 years through CanCogen, but also major investments in the provinces, major investments by the federal government through the Public Health Agency of Canada. And so that because of that, what we've got now is capacity across the country to really be able to do the work, to do the sequencing. Do we need to be doing more sequencing? Maybe, but I don't know that it's as easy to say yes across the board. I think here and there in different places, we're still building out capacity, we're still building out throughput. I think what is the most important is having the both hard and soft infrastructure to be able to modify based on kind of what's happening on the ground, up and down, the ability to incorporate new ways of tracking, like for instance, wastewater that Catalina also mentioned, which you can track kind of not just the quantity of virus in wastewater, but also potentially variants. I think it's also really important for us to be thinking about the sustainability of that system going forward. And that's something that certainly the National Microbiology Laboratory, the Public Health Agency, and others are building out. And I think that needs to be our focus in many ways over the next couple of years, is how to build a sustainable system that will be able to be in place and can be ramped up or down depending on circumstance. And then we need to work on smoothing out some of the information flow. And so it's not just about doing the sequencing work, it's about getting that data into the hands of policymakers, but also researchers and global collaborators much more efficiently. So to me, it's a more nuanced question. It's a great question. It's not just about the actual throughput, it's about all the pieces that kind of go around it that I think are really crucial.
Yeah, and maybe I can add a couple of comments. Rob, you cover really almost everything, but a couple of comments there. And one is that since CanCogen was the beginning of CanCogen, we already conceived these as a project that will be an initiative that will be transferred, and the capacity that will be transferred directly to the National Microbiology Lab. And I think that's an important aspect. And when we compare with other initiatives across the world, we think about, if we think about COG UK, that has been the leader on how they use genomics during the pandemic, they did all their effort based on academic labs. We did our effort based on public health labs. We built the capacity already in public health labs. We spent 2 years building the capacity, training the people, getting the equipment, getting the throughput, getting the analysis, everything in the provincial health labs, decentralized across Canada, and also part of it centralized through the National Microbiology Lab. So that's really, I would say, a little bit of a game changer in how we are preparing for future pandemics in Canada and for future use of genomics, not just for pandemics, for any pathogen surveillance. Think about food pathogens, other pathogens, HIV surveillance, tuberculosis, all kinds of different pathogens that are present in Canada that are now, we will be able to track them much better because of the capacity we have built in genomics in this country. So I think that's, again, setting a whole new precedent for us. As Rob mentioned, is not as about how many sequences we generate, but how we can use this capacity across the country to be really better prepared. I couldn't close here without mentioning again and again and again and once again data sharing, which Rob mentioned, but is really the critical component. And I think that's where Canada should really, I think that's going to be our major barrier and roadblock in the next years. And that's where we should put really important efforts. I think, again, a game changer could be if we were able to share the data in a much faster and meaningful way. And we show a little bit of that with Cancogen, with our VirusSeq data portal. We created the portal to share the data. And I think that creation of the portal is paving the way, not just for pathogen surveillance data sharing, but for health data sharing in general. And again, that's going to be critical. It's not only the bioinformatic analysis, but it's the capacity to aggregate the data and analyze it all together with powerful bioinformatics tools, but really having access to the data is critical.
Thank you. The next question is from James Tiet. What are the findings from assessing human genes and severity of COVID? I have heard there is an international effort for a larger data set for determining these associations. Is Canada part of that? And when do we expect substantive insight?
Yeah, maybe I can start briefly and let you, Rob, add to that. But yeah, as we mentioned at the beginning, there are two components to Cancogen. One is the viral component where we sequence the virus, and the other one is the human component where we sequence the host, the humans that are affected by COVID. And then we look for the difference. There's a lot of work that's being done in that area, finding genes that make people more susceptible to develop a more severe disease. Also looking for genes that are associated with vaccine response or drug response for treatments. We have here, one of our participants is Vincent Muser, that has been leading the Quebec Biobank. And they have been working with us and with Navid Aziz and others at CGen and with us at Genome Canada to advance this initiative. So what we're doing here is that for HostSeq is a project of projects, basically. We are collecting samples from projects that are already working with COVID patients, and we are sequencing those, and we are collaborating with the UK, with the US, and aggregating our data to all these international data banks. So there's several publications that have been issued from this work where the sequence have somewhat enabled this research that's being done in the susceptibility humans have to have more severe disease, or in some cases the resistance. Why some people get exposed to the virus several times, and you have a family where everybody's super sick and one person is positive but not really, or basically asymptomatic. So all these are answers we're working on our core for HostSeq. As I mentioned, we have more than 7,000 samples already sequenced, and we are now actively with international partners analyzing that data and making sure that data is also part of international consortiums.
Any additional comments, Rob, from you?
No, in the interest of time, I think we just keep rolling through these questions.
That's great. Thank you. Next question, Nate Rain. And can you share your opinion on genomic sharing initiatives in Canada? I think it's in line with the previous question. CanDig, CanPad, etc. And whether there are new opportunities and initiatives in 2022, Genome Canada will be funding to promote further clinics' genomic data sharing, in particular programs similar to the UK's Genomics England and or NIHR biorepository.
Yeah, maybe I'll just to say, I asked Catalina to speak to this, but just to say that this is a great question. We do have, unfortunately, a bit of a fragmented system in Canada when it comes to the variety of cohorts that exist. We actually undertook a project with CIHR to kind of do a survey of those cohorts and then to look at kind of ways forward. And we will be addressing kind of the results of that work as we come out of the kind of the COVID focus. And in particular, I think it won't surprise anyone that one of the key findings was around equity and inclusion and the need to make sure that the data sets that we're developing in Canada when you put them together, are going to be representative and allow for perhaps a bit of a more accurate picture of those who live in Canada. But Catalina, do you want to speak maybe a little more specifically to that?
Yeah, thank you, Rob. And as I mentioned before, data sharing is an important challenge, not just for Canada. In Canada, we have, of course, when we talk about health, we have a provincial healthcare system that is fragmented, and that makes more difficult for us to share data across the country. But this is an international challenge. This is really a challenge that really, again, is not only for Canada. And what we are working, and again, one example is the VirusSeq data portal we launched last year, and that is helping just share the data across Canada, keeping all the security and privacy parameters. So it's important to make sure that we, look at the security, look at the privacy, and develop a framework where we could have federated data that is connected. It's not about putting all the data in a library and getting everything together, like, the simplistic way of thinking about data sharing, put everything in a pod and everybody access. Here is more like federated data, because we are a federated system, and giving access and connecting the dots. And that's basically what we are also trying to do with HostSeq when we are accessing human genomic data with clinical information through HostSeq. So we have the two examples now, the one that we are putting forward with HostSeq with human data, and the one we put forward with the virus. And we're also thinking actively and proactively on how we can start sharing data. Just to give an example, we have now actually 4 projects that are funded around biodiversity that are collecting data, sequencing diverse organisms in Canada. And we're thinking, how can we bring this data together? How can we aggregate this data? And hopefully that would be less sensitive in terms of security and privacy than the human data that we're always so worried about the implications and privacy and all that. So those are things that we, as part of our mission-driven work, we are very actively thinking about and proactively looking for solutions for our data in Canada.
Thank you. The next question from Susan would go on. I think we have already covered these questions in the first section of the discussion, but in case if you have any additional comment, Rob and Catalina, Susan is saying, can you talk a bit about how the great research from Genome Canada efforts influences policy decisions? Once again, I think we talked about this, but if there are any additional comments.
Sure, I'll just make a general comment. Thanks, Susan, for the question. So we talked about COVID, and we talked about the ways in which this is informed decision making. So we don't need to go over that. Of course, the same types of information can help with decision making around, say, for instance, rare diseases, when we have better diagnostic tools and so on, right? The one thing I will say, just as a general observation, is Genome Canada from the very beginning has included as a key element of all of our major research projects what we call GELS research, right? So this is genomics and its ethical, environmental, economic, legal, and social aspects. And this research is really meant to go beyond just the technology and the science and to start looking at like implementation and application. And so you often on research projects will have health economists, for instance, evaluating the economic impact of new technologies so that there's, it's easier for policymakers to decide whether or not to adopt it. Projects in forestry, for instance, might be working with sociologists who actually speak with foresters to determine kind of the social acceptability of new tree strains and so on. So the incorporation of really important social science research into broader projects that involve technology and science is a key component of providing, I think, usable information for policymakers more broadly.
Thank you. Catalina, any additional? No, okay. That's
Nothing
good.
else to add. Rob did a good job.
Okay, the next question from Jean Saintville. How do you propose we optimally manage concerns related to data security, geopolitical considerations, global conflicts, etc., while there is a need and desire to conduct international collaborative research? I think that's an important question that it most of the people's minds. So please, who wants to take this first?
I'll say a couple of quick words. So, Jean, like that is like, thanks for the very simple and straightforward question. This is the fundamental tension, and this isn't the genomics issue. This is a science issue, and being a science policy, panel and community here, I think this is something that we will all be contending with over the next 10 years, maybe longer. We've had, I think, a period in which globalization and a sense of global collaboration has really been kind of on the rise. It's been seen as generally positive and has been kind of increasing, but borders are thickening. Concerns around security, data security in particular, are rising. And we are all going to be struggling with the balance between open science and shared and collaborative work with what we do see as a changed international relationships and realities. Just at a very, very high level, I think we need to be thinking, we can't lose sight of the openness and collaboration, but we need to take these concerns into account by doing things like, for instance, perhaps Carolina mentioned kind of the aggregation of data. So we, inside our own borders, perhaps we can do a certain amount of aggregation, perhaps, even more kind of anonymization before submitting to international databases, for instance. We need to be thinking about ways, even internally, we think about ways to adopt federated approaches to analytics. So instead of the idea, everybody just put your information in a single repository, we need people to be able to retain control over their own information, whether that's at the national level, the provincial level, or even the individual level. But we can distribute the analytics. So it's actually the algorithms that go out and find the data, rather than sending all of the data to somebody else who now in effect has control over it. So I think there's some specific things we can do, but I actually think this is a fundamental question that all of us will be dealing with for many years to come.
Yeah, and I don't have that much to add, just to say that, again, we're working with the federal government. We're working also with the province and with the genome centers in looking at the different aspects of data security and global security. And as Rob mentioned, this is not genomics. This is a science and research issue. This is a global issue. This is a geopolitical issue. And we are very actively looking into that.
Thank you. The next question is from Casey Hubert. Once again, my apologies if I mispronounce your name. Catalina, you may want to go first for this question. Are the 330,000 SARS-CoV-2 genomes that CanCoGen has sequenced so far all from clinical cases in Canada? And for policy development, do all viral genome sequences have equal value? International data sharing is great, but should Canada be investing in its genome sequencing dollars abroad? Where are the genome sequences out of South Africa last fall the most valuable genomes for policymaking everywhere, including in Canada? So these are very, important questions, bringing different dimensions of, investing in international collaboration. Catalina, over to you.
Yeah, thank you, and thank you, Casey, for this question. So to begin with, the 330,000 sequences we have are all from basically people that are in Canada. Some of them might be samples that are taken as part of our strategy for border control. So basically there's border surveillance, I will say, doing genomic surveillance at the border for people that are coming to Canada, that are potentially traveling from other countries and that get tested. But that's a smaller number as compared to all the surveillance we're doing across the different provinces from people that are affected by COVID and are being tested, are positive. And we have some selection criteria to decide which of those samples, the positive samples, the millions of positive samples we have in Canada, or thousands of positive samples, which ones are really sequenced. Now, in terms of the value and equal value, I think is an interesting question because, of course, every genome has an important value. But for decision making, we just to give an example, when we have an outbreak, for instance, with 100 individuals, we don't need to sequence the 100 individuals to get the information. We might be able to sequence only 15% of those 15 individuals or 50% of an outbreak to really get the information we need to make policy and public health decisions. So it's not really about sequencing a lot and everything. is about sequencing with strategy and defining the sampling priorities. And this is not just Canada, internationally, because of course, sequencing is expensive. It's not just the value of a PCR test. It costs more per genome. And it's important to make sure that we give use to those sequences. Now, in terms of the South Africa case for Omicron, I will just make a comment here in terms of the equity and in terms of some of the global challenges we have around genomics. If you recall, that finding of Omicron led to lots of travel restrictions to Africa. And then we figure out later that Omicron was present in the Netherlands way before. And now we figure out that Omicron was present in the US way before South Africa. So I think there is also an important role that Genome Canada and other international leaders have to play in making sure that data sharing is not penalizing any countries. And I know the World Economic Forum, the World Health Organization are actively looking at those questions, and we're part of those forums discussing at a global level how genomics can have an impact and how can we use this data in an equitable, inclusive way for the benefit of all.
Rob, any additional comments to me on that?
That's a good question. That's an important question.
Thank you. Next question from Mark Samuels. Aside from the immediate concerns about the pandemic, the greater threat to our society is climate change. How can genomics help with this problem? Rob, you may want to take time.
Yeah, so excellent. Thanks, Mark, for the question. And first, now just a shameless plug to go and read the editorial that was published at the CSPC site, because that's really what we focus on in that editorial, which is, and just as a little, maybe a bit of a sneak peek, we're almost certainly going to make, climate change a major focus of our kind of next round of funding. And because we understand that this is, of course, a great challenge, again, facing all of us around the world. And genomics has a role to play. So there's lots of ways in which genomics can help, both in terms of getting to net zero carbon, in terms of adapting to the changes that are happening. And that crosses sectors from agriculture to natural resources and energy, thinking about ways we use fertilizers, for instance, mitigation measures. There's any number of ways. So, it's very much front of mind for us at Genome Canada, and not just at Genome Canada, right across the country. We work with genome centers in all the provinces and regions. This is a huge focus for all of them, as well as internationally. So absolutely a focus for us.
Can I add just one small short comment? And as Rob saying in the editorial, that the link is on the chat. You will see some of the examples on how genomics is helping us understand, adapt, and mitigate climate change. But I want to use also this opportunity to answer the question that is below from Kate Orvis on the genomics in the mining sector. That I know we always get people asking, how come mining is about minerals, and what is genomics doing in the mining sector? And actually there is a big role for genomics, and I will say three areas where genomics has an impact in, and has a potential to have an impact in the mining sector. First is water management, and as you know, for those of you somewhat familiar with the mining sector, water is a big challenge for mining companies. Managing the water, keeping the water clean, and monitoring water with biotechnology tools and with genomics is a big component. The second, bioremediation of soils. As soon as sites are left, we need, as part of the regulation for mining companies, you need to do and go into bioremediation of those sites. And genomics, big time, with all the bacteria and all the different components that genomics can bring, could help with this bioremediation. And the last one is, of course, increasing extraction yields by using biotechnology, green approaches, and genomics. So those are three areas where concretely, and again, linked to climate change, because of course mining is having an impact on our climate in water and bioremediation. So all those somewhat linked directly also to climate change.
Thank you. Before going to the last set of questions, I want to ask my colleagues to share the one last poll, and that's a one-question survey from our end, and two participants to let us know how we performed. We always rely on your feedback, and always feel free, please, to reach out to us and provide suggestions, ideas, and criticism. Even we would love to hear from you. Thank you if you answering the questions. Going to the next question from, I think, Baja Paddy. Antibiotic drug resistance, AMR, is a major cause of death in hospitals across the world. What is Genome Canada's vision on this aspect? Anyone who wants to?
Carolina, why don't you kick off with this one?
Yes, absolutely. So thank you, Rob. AMR is indeed, there was an article on The Guardian yesterday saying that it is actually now our biggest challenge in the world in terms of health. And we absolutely are now thinking. We have been organizing, in particular, Genome Alberta has been very, very active on the area of AMR with international leaders, in particular the UK, that has invested several million pounds in using innovation and research to address challenges as AMR. Some of the things that we're thinking about is the tools that we are now developing for pathogen surveillance and for biomonitoring. The ones we developed for Cancogen, for COVID-19, as I mentioned, the wastewater tools to monitor antibiotic presence in waters, in agricultural regions, and monitoring also AMR in animals. And that really lead us to one concept that we are exploring very much, and that we're putting forward, and we'll be hopefully making at one point some announcements on potential funding around these, but is really around One Health. So it's not just, and we saw it with COVID, it's not just about our human health, but it's our interactions with animals, with the animal kingdom, with plants, with the environment, with water, with all that. So bringing that concept of a more holistic view of health and a more holistic view of our analysis of pathogens, and how those, some of our misuse of antibiotics is leading to this great pandemic that we call pandemic, we could call pandemic also, which is the antimicrobial resistance. So very, very important topic for us. And we, again, are actively looking at the role genomics is playing. We do have some projects on that have been funded in that area. And we're now looking at potential future funding and collaborating with other agencies in Canada. The federal government is also planning important investments around AMR, and we are aligning with those too.
Okay, that's good. And there is also a comment by Mark Perry. I read the comments, but I'll go to the next question. And in case if you want to combine your answers or feedback on this, comment is, your Canadian tax dollars at work building out public health lab genomic sequencing for the next pathogen, +1, infrastructure investment equals improved capacity. So that's a comment. But the next question, and feel free to reply back, Rob and Catalina. The next question, however, is about the fragmentation from Karen Haiti. Rob mentioned fragmentation as a driver for genomics. From the research perspective, could you elaborate on the nature of this fragmentation?
Sure. Yeah, happy to do that, Mehrdad. And I know we're almost at time, so I won't belabor the point because it's a big question. And frankly, it's not just a genomics issue. Maybe I'll start more broadly. I think COVID brought home to us that we have just enormous strength in Canada when it comes to research, when it comes to public health, when it works to comes to people in the hospitals, but coordinating the activity across the ecosystem proved to be immensely difficult, and that's in part because of the way our federation is structured between federal and provincial governments, even within provinces. That some provinces have a multitude of public health units that all have their own privacy officers that need to sign off on things. But then even at the federal level, we have, for lots of understandable reasons, we have different funding agencies that do different types of things. Health in COVID transcend. So it's not just a CIHR thing. It's not just, there's Genome Canada's involved. You have CFI with lots of infrastructure and so on. NSERC's doing a lot of work as well in this space. And then you've got, of course, got Government of Canada, research labs and so on, and industry. And so when you need a coordinated approach, and a lot of these challenges do require a coordinated approach, how do you bring together a community to try to, to move things forward, whether that's in response to COVID, whether that's in response to climate change, whether that's even just in response to improving economic conditions in Canada, getting government, academia, industry on the same page, at federal level, at the provincial level, individual labs and universities and so on. That coordination is difficult. And we see that in genomics. There's no doubt immense strength across the system, but getting the pieces to work together really efficiently is something that we need to think about going forward. And I'm convinced that that this is on not just our mind, but on the minds of many others. So something we'll be tackling.
Thank you. And Catalina, any final comment from your end?
Well, maybe not specifically on that question, but maybe in general, I will say that, I think we are really, we have been talking about the genomic revolution for a while, and I think we are now really in the midst of that genomic revolution. We have the opportunity now to address many of the challenges we have, and we have discussed today on being the climate change, the AMR, pathogen detection, even precision health. I think it's a unique opportunity for us, as Rob mentioned, to work collectively and to really align our efforts. And I would say that if we take one example, the one in the UK, I know they have many, many challenges, but they have been able to align industry with academic groups, with government. That I think that three-part interaction is critical for us to be able to deliver on the big impact of genomics in Canada, with these interactions between the private sector, the academic groups, and the government.
Thank you, Catalina and Rob. So we were able to answer a good number of questions. So we did relatively well in that case, but I think there is still tremendous amount of questions and discussions remain to be discussed and answered. So I think we have to take a rim check and invite you back, Rob and Catalina, maybe in 6 months from now, to again reopen this conversation and discussion, perhaps post-budget, and then see how we can perhaps expand the conversation on genomics on a mission. Keep the conversation going using the hashtag SciPol series and Genomics on Mission. This brings us to the end of this conversation for today. And I want to thank my colleagues at Genome Canada for their excellent collaboration and co-hosting this session with us. I want to thank the participants and being engaged in the discussion. And finally, thanks, Rob and Catalina. It's always great to have you with us and sharing your insights with the audience. We're very much looking forward to continue this conversation. And one last point, if you are not registered for CSPC newsletters to be informed about the upcoming events and other happenings, please register for CSPC newsletter. Thank you so very much, everyone. Merci beaucoup à tous. Bon après-midi. Bonne soirée. Thank you.
Thanks, everyone.
Merci à tous.